Hello, good morning and good afternoon. We'd like to welcome you today to the Virtual Pharma Symposium hosted by BASF Pharma Solutions. I'm your host and moderator, Frank Romansky. I'm the Head of Global Marketing at BASF Pharma Solutions based in Florham Park, New Jersey. BASF Pharma Solutions has developed this free series of webinars this week as a Virtual Pharma Symposium. We welcome your questions during the seminar using the On24 platform, as well as follow-up requests on any of the topics presented here. BASF Pharma Solutions focuses on excipients, APIs, and ingredients in a number of segments, namely solubilization, instant modified release, soft gels, biologics, and skin delivery. Today we will focus on the skin delivery element with our in-house expert Benjamin Goodyear and our academic collaborator, Professor Murthy. So let me walk you through our agenda that we're going to have today. First, Benjamin Goodyear is going to present modifying API permeation through human skin with lipid-based oleochemical excipients. It's going to run for about 30 minutes. Benjamin Goodyear is a Global Technical Marketing Manager for BASF Skin Delivery Portfolio. He leads a virtual platform team of technical experts that engage with topical formulators focused on generating application data used to help topical formulation product developers. Ben has worked extensively with dermatologic relevant portfolios for GSK Consumer Health, Teva Pharmaceuticals, Amneal Pharmaceuticals, and L'Oreal USA. Currently, his global team drives innovations within BASF's skin delivery platform and development activities for RX, GX, and OTC applications. His academic PhD training in pharmaceutical sciences at Rutgers University has helped him collaborate with numerous universities and industrial partners. After that, we're going to have a presentation by Dr. Narasimha Murthy from University of Mississippi. He's going to present Using Lipidic Excipients to Enhance Drug Permeation in Topical Cream Products. This also will go for about 30 minutes. Dr. Murthy is a professor of pharmaceutics and drug delivery and associate director of the Center for Pharmaceutical Technology at the University of Mississippi School of Pharmacy. Dr. Murthy is also the founder director of a philanthropic, nonprofit research organization, the Institute for Drug Delivery and Biomedical Research in Bangalore, India. Dr. Murthy's research programs are funded by the NIH, the FDA, and numerous pharmaceutical industries. He has published over 120 research papers and presented over 200 scientific posters in various national and international scientific meetings. He has authored 2 books and 15 book chapters. Dr. Murthy is also the recipient of numerous industrial and academic awards, and we would like to welcome him today. Following the presentations, I will host a Q&A session where we will collect your questions and answer them as they come in. So with that, I'm going to turn it over to you, Ben, and allow you to start in the presentation.
Thanks, Frank. I'll be talking about modifying API permeation through human skin with lipidic-based oleochemical excipients. And then we could stop, right? So today I'll be covering the following topics to prepare you guys with some important chemistries involved in dermal drug delivery. We know it's important to consider the penetration pathways and mechanisms of dermal drug delivery in order to effectively deliver APIs and excipient permeation through the skin. We also will review some of the methods to measure critical quality attributes and see a few supporting examples of permeation testing and spectroscopic evaluations. The human integumentary system makes up approximately 10% of your entire body mass. It's the largest organ on your body. The uppermost layer of the skin consists of a lipophilic stratum corneum, approximately 15 micrometers in thickness. The stratum corneum is incredibly efficient at providing protection against foreign substances, in this case, API and excipients, due to its unique diffusion gradient caused seen in deeper, more hydrophilic layers of the epidermis and deeper layers of the dermis. A comprehensive understanding of drug and excipient interactions with each other is advantageous to overcoming drug bioavailability changes commonly associated with topical and transdermal drug delivery. Many of us are aware that there are several mechanisms that facilitate passive drug diffusion for localized or systemic drug delivery. 3 common routes are through the skin and are shown in the first chart on the left. The blue is highlighting the hair follicle. This is the first route. The second is the red arrow, which is showing the sweat pores. And the green arrow, which is the smallest one there in the left figure, which is the stratum corneum. We will focus for today on the stratum corneum for today's symposium. And as you can see in the center chart, the stratum corneum has 2 important pathways understood for effectively delivery of both hydrophobic and hydrophilic drugs. The hydrophobic APIs, so this is the green arrow in the center chart, chemistries tend to favor the lipidic pathways. The hydrophilic pathways, which as you see that long red arrow going through the corneocytes, favor their intercellular pathways. Many skin models have been developed to try to mimic these unique skin properties, but due to the highly complex, organized lipid lamellar structures, making these models is just not so easy. Physicochemical characteristics are known to impact drug permeation into the skin. Rate of diffusion, or flux, is affected by these various parameters that you see on the chart in front of you. We've got API melting temperature. Obviously, it's best to have less than 200 degrees Celsius because of the state of the drug is going to be more solid. API polarity, hydrophobicity or hydrophilicity, it's obviously best if the log P value is within the range of 1 to 3. API molecular weight, so for example, the size, it's best to be under 500 grams per mole. And your aqueous and solubility, pH, and charge, there's also some properties that are favored as well. So frequently asked questions formulation scientists have for excipient suppliers are related to the specific dermatological drug API characteristics. So all of these are many of these common questions that we have to see how our drugs will work with excipients. And having information on these parameters can help us quickly pinpoint which specific characteristics are most noteworthy to pay a close attention to. So at this point, we've talked quite a bit about the drug's physiochemical properties. How about the excipients that make up the majority of a semi-solid dosage form, right? We first need to consider the potential interactions that there might be on drugs' ability to penetrate the top layer of the skin, the stratum corneum. So number one, the drug itself. Depending on how much drug loading you have, you could have between 1 or even 10% that you use in a formulation. There is a thermodynamic driving potential that might increase when more or less drug is loaded into a dosage form. Secondly, healthy skin versus diseased skin is another common consideration. You might show better skin penetration with adequately hydrated corneocytes, and there's more ability for that formulation to penetrate through. Thirdly, you could consider pre-treating the skin with organic solvents or toners, if you will, and that might disrupt the barrier integrity of the highly organized lipid lamellar structure of the stratum corneum, which thus results in increased drug permeation. Or if it's not one of those routes, additional associated ion pairing and vehicular complexation may also alter the chemistries and the nature of the API itself. So here's where we will briefly point out some known concepts behind skin penetration enhancers. Regulatory has seen some major hurdles and challenges as there's no one-size-fits-all when it comes to comparing the behavior of each one of these different chemical penetration enhancers and what that effect has on the dosage and the variation of the dosage form. There may be actually several penetration mechanisms we can link back to either the drug or the formulation vehicle from what we know in the chemistries. And one could actually quantitatively measure a significant increase in cumulative drug permeation or flux using a lower viscosity formulation. But for now, it's best to avoid comparing across these technologies that are just microstructurally different until we kind of focus on some of the chemistries involved with the enhancers themselves and their role on chemical penetration enhancement into the various layers of the human stratum corneum system. So hopefully at some point in your journey into topical formulations, especially with creams, you had a chance to understand two, major classifications important to giving key benefits to the dermal drug delivery and efficacy of APIs into the skin. So this chart in front, we have emollients on the left-hand side and structuring agents on the right-hand side, categorized into two groups. And some of the key the benefits of dermal drug delivery with emollients are that they're very lipidic in nature, and they contain these double bonded oxygens. And this added bond energy may have some additional impact with hydrogen atoms. Others contain ketones, and in the case of octadodecanol, one can expect that it is a hydrolysis-resistant chemical due to its chemical nature. And these will all be liquids at room temperature. And one thing to also note here is that on the other hand, on the right-hand side, you probably need to spend a reasonable percentage of these structuring agents or various fatty alcohols on the right-hand side of the chart here. So in most cases, you'll typically see these blended together very nicely to take advantage of some of the working saturated fatty alcohols you might find that are, some that are liquid, some that are solid, but it's these mixtures of these different fatty alcohols that can effectively help us tune and optimize these semi-solid dosage forms, much like how we see in creams for these, for example, structuring agents. For today's talk, we're interested in exploring some of these properties in the labs and some of the data that we're going to be showing, with the most promise for these emollients on the left-hand side as chemical penetration enhancers. So why do we care about the materials and the methods part so much as pharmaceutical scientists? This is a question I've asked myself a lot, especially recently. And because you have to have good materials and good process, and hopefully you'll have a good finished product at the end, right? Like, how are we supposed to even prove this? Is, good in equals good out. And for example, the most important part of your experimental design really needs to consider the fact that critical quality attributes can actually help formulation scientists, which are the most relevant to your dosage form. So if you could be working on a cream or a foam or a gel, there might be different CQAs that you might be interested in exploring, especially if you know the mechanisms in which your drug is delivering into the skin. So for these examples, some of the CQAs that are definitely noteworthy here are the API solubility, the microstructure, which is the viscosity or the stability of that formulation, and maybe some sensory aspects, because those are some of the most important things for sensory you might see in a clinical setting. And there are definitely some noticeable differences when you go from bench to pilot and product commercialization from start to finish, which is why it's so important to define these CQAs when you're formulating a topical dosage form. So here we have a gold standard Franz diffusion cell. Most of the people here on this call will probably know what this is, but for those of you that don't know what this is, I'm going to just review a couple of the major components of what this Franz diffusion cell does and how it works. So these major components that you'll find in the FDC are the donor compartment, which you can see in the graph on the right-hand side at the very top, almost like a cylindrical shape. The formulation or the dosage form is then placed into the donor compartment chamber, which is affixed to a membrane filter. For example, if you're running IVPT, it will be human cadaver skin. But if you're using one of these IVRT models, either cellulose membranes or a Strat-M is suitable for many applications. Although it doesn't have all the components of skin, it's still used in a lot of these high throughput studies. So if you go past the membrane and into the receptor chamber, which is towards the bottom right of the diagram, you actually have a little magnetic spin bar that will spin at a pre-designated RPM, and you'll have a circulator jacket that incubates the donor or the donor and receptor compartment to meet the specifications of body temperature. So in many cases, you'll have 32 degrees Celsius. If it's in vivo, it's going to be 37. But depending on your dosage form and what the goal is, your temperature could vary. So here's two graphs that show and demonstrate the differences in cumulative drug permeation for clotrimazole between various fatty alcohols, which is shown on the left. You have cetero alcohol, cetostero alcohol, ceto-alcohol, and stero-alcohol. And as you can see, the time point is between 0 and 8 hours. On the right-hand side, you've got some of these lipidic emollients. And for example, these are lipidic emollients we tested here were isopropyl myristate, mineral oil, Polycream 3C, and we also have tried just regular standard model drug without any type of enhancer. So what we also aim to do in these studies is take a look at just the overall differences between the fatty alcohols and the lipidic emollients. So as you can see on the left-hand side, we have the fatty alcohols, which kind of range at 8 hours between 100 and 140, the cumulative drug permeation. Whereas on the right-hand side, when you're taking a look at the lipidic emollients, you have a very large range. This is ranging from where you see the green from 60 all the way up to 200. And from these two charts, I mean, you can just clearly see like why lipidic emollients can have such a huge advantage on the cumulative flux or the cumulative drug permeation in this example. And some of you might be wondering why 8 hours. And the reason why in this particular model it's 8 hours is it's actually dependent on the dosage form. So 8 hours is indicative of having maybe 2 or 3 applications daily. So 8 hours in this case is actually sufficient. And others actually require testing every 6 hours. That would be in the case of, let's say you would have a 4 times daily dosing. The total amount, I mean, this is another question that comes up, is the total amount of fatty alcohol content that was used for these formulations was approximately 7% on the left graph. And on the right graph, the total amount of lipidic fluid that we had used was approximately 12% in all of these cases. So, with that said, I mean, there's a clear difference between fatty alcohols as helping with penetration enhancers and lipidic excipients, as they have a marked increase in cumulative drug permeation in this example. So what's ATR-FTIR? Attenuated Total Reflectance Fourier Transform Infrared Spectra. Try to say that 3 times fast. Or you could just say ATR-FTIR. We use this really neat device to explain a little bit more about the methodologies of testing chemical penetration enhancers. So actually, what you're seeing in this chart here is the blue, the plaque, and there's also a gray line. And from what you can see circled in red, the wave numbers on the x-axis. In this case, you won't be able to really see it on your screen, the wave numbers that are here. But essentially, each one of the wave numbers located on the x-axis is giving us different chemical signatures. This is essentially the unique fingerprint that a lot of these molecules have when we're looking at APIs and excipients. To be able to understand what that fingerprint is correlated to will actually help us with being able to image in more details. So looking at the signatures in more details, this is how we can see what the chemical interactions are. And it's important to note that this technique is only semi-quantitative, and it may not necessarily give us a sensitivity required to differentiate all the different chemical fingerprints associated with the enhanced vehicle or the enhanced API or the difference in skin interactions. But what we can do is we can determine which wave number actually corresponds to which enhancer. And this information is super important to help us determine the impact of barrier disruption when looking at the different intensities. So this is what lipid disordering looks like. Lipid disordering can be shown in the following way using FTIR imaging techniques. So based on that color scale that we have in this photo and the signature that we used from the recent test that I showed you, the FTIR-ATR, The color scale is where you see the blue is actually where least amount of intensity is. And red is obviously the most, and green is right in between. So the image provided shows that the lipid disruption of Cream 3C using a standardized sample preparation technique, it has some clear differences. So the image that you see, there's two boxes on the right-hand side. We have control, deposition, and layers 1 through 6. And based on the method, obviously we didn't show layers 3 and 5 because we wanted to highlight, the deeper penetration of the various lipid disruption in the skin. So a lot of people will ask us that question. But from what you can see with 3C, there is a huge amount of disruption, which is the first one, a lot more red. And the formulation without 3C, we see much less disruption, so it's more blue, which means that the lipid lamellar ordering packing is less disordered. So here I'll just break it down a little bit further for everyone to kind of understand what does this mean. So what does this really mean? So we have kind of three figures here. We've got a placebo formulation, we've got a formulation with lidocaine, and a formulation with clotrimazole. So placebo, lidocaine, and clotrimazole have all been tested using the exact same methods. And we can share those methods with you, but for the sake of this presentation, I just wanted to show you what this really means. So in the first graph where we see the placebo, what we're tracking right now in red, as you can see, from the deposition to layer 1 all the way to layer 4 is very much disordered. And when you start going down to layer 6, it's a little less disordered when you have a formulation with Cream 3C. And if you compare that to the control without 3C, there's a huge difference. On one end, you're seeing a lot of red and very, very deep penetration for this placebo formulation. We're looking at 3C. And without it, you don't see any 3C. So that's why we just see blue, is because we don't have any 3C in the formulation. So the placebo one was tracking 3C. And then the second graph, we've got lidocaine. Lidocaine, showing the effective permeation into layer 6, just as 3C did in the first chart. So the effective lidocaine permeation versus the formulation without 3C, there's a clear difference. And finally, in the third graph with clotrimazole, we're obviously tracking here with and without 3C. We can actually see the drug permeation go much deeper into the layers of the skin. So this was super interesting because without it, you're not even you're getting a dark blue. You're not even making it into the deeper layers of the skin. So this is definitely something noteworthy to explain, that the disruption that we see in the first chart is also something we're seeing with drug formulations into the other charts. So the idea here is that 3C is helping with the cumulative drug permeation into the deeper layers of the skin. All right, so switching gears here. Now let's briefly take a look at another spectroscopic imaging technique called confocal Raman. And in the graph here, you can actually see the device and how you would, theoretically place it onto the surface of the skin. This is a non-destructive technique that actually doesn't really impact you negatively. So that's why it's so convenient, is that you don't need any other special equipment other than the actual device itself. And obviously someone that can read what these charts are saying. So today I'm going to actually go ahead and kind of show you what that looks like, and help you interpret the dermal drug delivery of a few things here. But let's, in this example that we're going to use octadodecanol for time and depth-dependent study. So clearly, we can observe just how deep the octadodecanol, the chemical penetration enhancer, gets into the deeper layers of the skin surface. So there's two graphs here that you see. So there's the one on the left and the one on the right. The one on the left is actually just showing the spectroscopic image after 20 hours pre-treating the sample in a Franz diffusion cell with octadodecanol. And we know that now, looking at this graph, that it's penetrating inside the stratum corneum until it reaches the epidermis using that chemical signature that we were able to determine in previous techniques. But we can detect the octododecanol up to 20 micrometers below the skin surface. So the chart on the left that you see is actually at 20 hours, the intensity of octododecanol, as a time-dependent study here. So as you go from left to right, you start seeing more and intense from bright red to like very dark red. So this is indicating that there's just more penetration into the deeper layers of the skin. And as expected, octododecanol can be detected in the untreated skin sample, which is the chart on the right. So in the spectroscopic images presented to the right, the higher the ratio is, the redder the image, and the higher the OD octododecanol concentration inside the ex vivo skin sample is. So using the first two markers, so like peaks at 1450 and 1300, we can actually detect the octododecanol up to 20 micrometers below the skin surface here as well. And using the last marker at peak 1080, we can also detect OD up to 15 micrometers below the skin surface. So this is super fascinating because, as expected, no octododecanol can be detected in the untreated skin sample, which is what we obviously expected. But reading this from left to right, you can see 10 minutes to 60 minutes with greater amounts of your drug product in the top layers of the skin that gets deposited or more drug deposition into the deeper layers from left to right. So that was the the interesting findings here. But to visualize these kinetics of OD penetration into the skin after acquisition of the repeated Z-line over an hour period, we contacted all the lines together to build these kinetic images. And these Raman images provided this kinetics to really help us understand these different markers. And we were able to use these markers to generate these images that you see in front of you. All right, so next slide here is a histological look at the human skin combined with this super fascinating FTIR imaging technique. So what you have here on the left side in the gray at the very top is the untreated sample, and directly to the right of it is its appropriate signature with using FTIR imaging. So we have untreated samples have a clear difference when compared to the triplicate measurement taken at 20 hours. So the top two are the untreated, and the bottom six are the treated. So from the left side, it's just the microscopic image that was taken, and on the right side, we're actually seeing the CH stretching. So octododecanol is actually showing a very intense red color at the very surface of the skin, and that's actually where we believe the stratum corneum also resides. And we know that this CH stretching is being caused by the addition of octododecanol, because when you go from the very top chart to the bottom three, you're clearly seeing a marked increase of lipid lamellar structure disordering. Well, there you have it, ladies and gentlemen. As promised, we discussed many important topics related to dermal drug delivery, barrier properties of the stratum corneum. We know it's important to consider the penetration pathways and mechanisms of dermal drug delivery in order to effectively deliver APIs and excipient permeation through the skin. We reviewed some of the qualitative and quantitative methods to measure critical quality attributes with supporting examples of permeation testing and spectroscopic evaluations. So I would like to actually thank our global technical marketing skin delivery platform team for supporting the preparations and the coordinations with our technical teams. It would definitely not be possible without them. And if you have any questions about my presentation here today, I would strongly encourage you to ask in the chat box during our Q&A session after the next presentation. And if for some reason we have a lot of questions that I don't specifically answer yours during the Q&A section, I would like to also have our next speaker have time to answer many of the questions that are coming up from them as well. But You can definitely send me an email with your questions, and I'll do my best to respond as quickly as possible. So without further ado, next, I would like to pass the ball over to Dr. Murthy so you may see some of his most recent research conducted at his topical testing facilities. Dr. Murthy, take it away.
Thanks, Frank and Ben, for the nice introduction. I am Narasimha Murthy, Professor of Pharmaceutics and Drug Delivery at the University of Mississippi School of Pharmacy. The objective for today is to discuss the use of lipidic excipients to enhance the drug permeation across the skin in topical cream products. I'm going to discuss about 2 different projects that we performed using the lipidic excipients in this presentation. Before I go into the details of the projects that we performed, I would like to briefly talk about the structure of the stratum corneum. Ben has already talked in detail about the structure of the skin. So generally, from the drug delivery perspective, we see skin as a bilayered structure. We have the outer epidermis and the inner dermis. The outer epidermis can be distinctly divided into 2 more layers. The outermost layer is the stratum corneum, and the inner layer of the epidermis is called as viable epidermis. The outer stratum corneum consists of dead keratinocytes, which are tightly packed, and the intercellular space is occupied by lipids. The lipid domains consist of the ceramides, fatty acids, and cholesterol. They are appropriately mixed together to form thermodynamically stable bilayer lamellar structures. Now it is very well known that the lipidic pathways in the stratum corneum are the major pathways responsible for the drug absorption across the skin. The chemical permeation enhancers, particularly the lipidic permeation enhancers, have been reported in the literature to be acting by multiple mechanisms. A few of the potential mechanisms are illustrated in this slide. One of the mechanisms by which the lipidic excipients could act as drug delivery enhancers is by the way of increasing thermodynamic activity of drug in the formulation. So the increase in thermodynamic activity of the drug would exert a driving force on the drug molecule, which would enhance the diffusion rate of the drug along the concentration gradient. The second potential mechanism is by the way of increased partitioning of the drug. So what this means is the drug is made to dissolve more in the lipid domains of the stratum corneum. Some of the excipients are known to penetrate themselves into the stratum corneum lipids and act as solvents to the drug molecule, thus enhancing the solubility of drug in the stratum corneum, which would eventually result in enhanced permeation of drug. Lastly, the most accepted mechanism and widely reported mechanism is the fluidization and disruption of lamellar lipid structures by the enhancers. So the fluidization and disruption of the lamellar organized lipid structures would lead to formation of aqueous pathways through which the poorly permeable hydrophilic molecules will be able to permeate across the skin and get absorbed into the deeper layers. The lipidic excipients in the topical cream products have multiple roles to play. They are used to improve the appearance, they're used to improve the flow properties and texture properties of the creams. Some have the ability to elicit some kind of a beneficial pharmacological effect also. What we are interested today is to look at the use of lipidic excipients as potential drug delivery enhancers in topical cream products. We worked on 2 different projects involving the lipid excipients. In project 1, we prepared topical oil and water type cream products using Colivax fatty alcohols. And in project 2, we again prepared oil and water type of cream products, but here we used the Coli cream lipidic excipients. Let us look at Project 1. The table on the slide shows the composition of the topical oil and water cream products that we prepared. So we incorporated clotrimazole as the model API. We prepared 5 different cream products with just one variable. Actually, the variable was a Colivax fatty alcohol. All the creams were formulated under the same processing conditions. So then all the 5 cream products were subjected to in vitro permeation testing. The barrier that we used was human cadaver skin, and we used transdiffusion cells for permeation testing. The tool for checking the barrier integrity was transepidermal electrical resistance. We used infinite dose condition for the permeation studies, and we used 2% tween in PBS as receiver medium to maintain sink conditions, and the sampling interval was every 2 hours. Let us take a look at the results of the in vitro permeation studies now. In the first graph, you can see the time course of cumulative permeation of drug across the skin. The flux was calculated from the slope of the linear fits. It is evident that the flux was different from different fatty alcohol products. Now we were curious to see if there is any relationship existing between the physicochemical characteristics of the fatty alcohol with the permeation flux of clotrimazole. Please take a look at the next 2 graphs in green. We found that there exists an inverse correlation between the clotrimazole flux and log partition coefficient and melting point of the fatty alcohol. What this means is that lower the partition coefficient value of the fatty alcohol and lower the melting point of the fatty alcohol, better was its ability to enhance the permeation of clotrimazole. There are a number of reports out there in the literature that shows that the fatty alcohols can penetrate into the lipid domains of the stratum corneum. So in this case, as the API is lipophilic in nature, any disruption of the lipid domains in the stratum corneum, any disruption of the lipid domains in the stratum corneum would only mitigate the permeation of API, right? Because the API is lipophilic in nature. Therefore, what we think here is it is likely that the lipidic excipient that penetrated the stratum corneum acted as a solvent or an agent that would allow the drug to form a reservoir in the stratum corneum. If the fatty alcohol is acting as a carrier or a solvent with the drug in the stratum corneum, then the drug should be soluble in the fatty alcohol, right? Not only that, the difference in the permeation flux in the permeation flux of clotrimazole that we observed across different products could also be arising because of the difference in the solubility of API in different fatty alcohols also. Hope you're following. So we decided to check the solubility of API in different fatty alcohols that we considered to prepare the topical oil and water cream products. We use DSC as a tool to determine the solubility of drug in fatty alcohols. What you see on the slide are 2 pictures, DSCs of different ratios of drug to fatty alcohol overlaid. In the first picture, we have 1:1 mixtures of drug and fatty alcohol. We did not see any endothermic peak in any of the fatty alcohols except SA. So what this means is all the drug that we incorporated into the fatty alcohols dissolved, except in case of SA, in which there was some amount of undissolved drug remaining that showed up the endothermic peak. So to know the actual relative saturation solubility of API in the fatty alcohols, we had to incorporate excess of API into the fatty alcohol. So therefore, we recorded the DSC of 3:1 API to fatty alcohol mixture we found the endothermic peaks appearing in all the drug fatty alcohol mixtures in the second picture. We also observed a difference in the enthalpy of fusion of drug in the fatty alcohols, indicating that there was different amounts of API remaining undissolved in different fatty alcohols. In this slide, we tabulated the enthalpy of fusion of API from the DSCs of 3:1 clotrimazole fatty alcohol mixtures that I showed you in the previous slide. On column 3, you see that the fatty alcohols are rank ordered in terms of their ability to solubilize the API. For example, Colivax MA, which showed least enthalpy of fusion for the API, was ranked first because it could dissolve a relatively more amount of drug in it as compared to other fatty alcohols. Then we wanted to find out if there is any correlation existing between the ability of the fatty alcohols to dissolve the API with the transdermal flux of clotrimazole that we observed in case of in vitro permeation testing of the topical creams containing different fatty alcohols. So needless to say that there was an exact match in the rank order that you see on column 3 and the column 4. So this supports our hypothesis that the fatty alcohol influenced the permeation of clotrimazole by enhancing the solubility of drug in the stratum corneum. That was about project 1. In project 2, we again prepared different topical oil and water type cream products. The composition of the cream that we prepared is shown in this table. The cream products are all of the same composition except one variable. The variable was the lipidic excipient. We used different cream lipidic excipients in the creams, and then we also used mineral oil as a control in one of the cream products. We prepared topical creams of different drugs, azelaic acid and lidocaine hydrochloride and metronidazole. Azelaic acid is relatively lipophilic as compared to the other two APIs that we studied. You also see the pKa values of the APIs that we chose for this project. We performed IVPT studies for all the topical cream products that we prepared. The IVPT studies were performed across the human cadaver skin. We used 2 donors, triplicate for each donor. The data that you see would be an average of 6 trials and standard error of mean. We use PBS as the receiver fluid. You'll see in the pictures the way we dose the skin. We basically use a syringe to collect the formulation from the container, place it on the skin, and apply uniformly across the active diffusion area. So one very important point that I wanted to make here is that all the studies were performed using infinite dose of formulation in the donor compartment, not finite dose, because in case of finite dose, a phenomenon called as evaporative metamorphosis occurs, so which leads to several changes in the formulation. Like it increases the viscosity of the formulation, it increases the concentration of drug in the formulation, the active diffusion area changes over a period of time due to evaporation of solvent and due to shrinkage of the formulation. Several such changes would occur in case of finite dose conditions. So that's why, to eliminate all those variables influencing the permeation of drug, we perform these studies at infinite dose conditions. I'm going to discuss the permeation profile, permeation data of different APIs from topical cream products one by one. I'm going to start with azelaic acid first. So what you see on the graph is the transdermal permeation flux of azelaic acid from different topical cream products. The second bar represents the transdermal flux of azelaic acid from mineral oil product. Obviously, Colicream 3C, Colicream OA, and IPM did not enhance the permeation, whereas Colicream OD significantly enhanced the permeation flux of azelaic acid as compared to mineral oil. So generally, when we see a difference in the performance of two products, we can either attribute it to the compositional differences between the two products or to the differences in the critical quality attributes of the two products. Right? The Colicream OD product and mineral oil product were almost similar in composition, except that they had one variable. The variable was the lipidic excipient. So before we attribute the enhanced permeation to the nature of the octyl dodecanol to act as a chemical permeation enhancer, we need to eliminate the possible influence of critical quality attributes on the performance of the product. So we characterize all the topical products for different quality attributes. First, let's take a look at the pH. You see that pH of the products did not differ significantly. It was clustering between 3.6 to 3.65. So the pH might not have played any role in determining the performance of the octyl dodecanol product better over the mineral oil product. Likewise, we also characterized the drug distribution between the aqueous phase and oil phase, and we found that the drug was predominantly located in the oil phase in all the cream products, and there was no difference in the amount of drug present in the oil phase across the products. So the phase distribution did not vary, and the amount of drug distributed within the oil phase did not differ significantly across the products. So this particular quality attribute did not play a significant role in determining the performance of octyl dodecanol cream. We saw from the phase distribution studies that azelaic acid is predominantly located in the oil phase. So in a oil in water type of dispersion system, if the drug is predominantly located in the dispersed phase, so we need to consider the globule size distribution as one of the quality attributes that could potentially influence the performance of the topical product. So that's why we went ahead and did microscopy and determined the globule size distribution of oil phase in all these products. So the D10, D50, D90 values are tabulated here. Again, we did not see a significant difference in the D10, D50, D90 values across all these products. There was some difference. The D90 values of mineral oil product was a bit higher. Otherwise, rest all were not really significantly different from each other. So lastly, we wanted to see if release rate across the products is different from each other. Particularly, we wanted to compare the Colicream OD with the mineral oil product. You see that the release rates were also clustered, and it was not really significantly different from each other. So now we have evaluated the pH. We have characterized the drug distribution in the oil phase and the lipid phase, oil phase and the aqueous phase, and we found that the drug is predominantly located in the lipid phase. And we also found out that the globule size distribution is not really significantly different across the products. And we found that the release rate was not really varying as well. So when all these critical quality attributes are not really contributing to the performance of the product or the difference in performance of the product, then what else can you attribute the enhanced absorption of drug from the Colicream OD product? Right? So it is definitely the ability of the Colicream OD to act as a chemical permeation enhancer, whatever be the mechanism. Okay, so it may be increasing the partitioning of the drug, or it may be increasing the thermodynamic activity, or whatever be the mechanism. There could be multiple mechanisms playing a role here. Okay, whatever be the mechanism, at least we are sure now that the enhanced permeation of drug in case of Qualitative OD product as compared to mineral oil was not due to the differences in the critical quality attributes. It was all due to the nature of the material, octyl-dodecanol, to enhance the delivery of drug across the skin. Right? Is that clear? Hope it is clear. Now let's take a look at the lidocaine permeation data. The graph represents the permeation flux of lidocaine from topical cream products containing different lipidic excipients. The last bar represents the transdermal flux of lidocaine from mineral oil cream product, which was our control. Compared to other lipidic excipient containing cream products, the Qualicream 3C product showed a significantly enhanced permeation of lidocaine across the skin as compared to a cream containing the mineral oil. Likewise, even in case of metronidazole cream products, you see that the last bar represents the control cream, permeation of metronidazole from control cream, and compared to any other creams, we saw a significantly enhanced permeation of metronidazole from Qualicream 3C product. Unlike azelaic acid, lidocaine hydrochloride and metronidazole are highly water-soluble compounds. So let us, like how we looked at the different quality attributes in case of azelaic acid cream products, let us take a look at the quality attributes here as well in case of lidocaine as well as metronidazole cream products. So if you see the pH of lidocaine cream products, they are all within a narrow range. Likewise, the metronidazole cream products had a range of 5.5 to 6 pH. And then generally, if the molecule is water soluble in an oil and water type cream product, the drug is predominantly located in the aqueous phase, that is the continuous phase, right? So in such a case, the globule size distribution has a very little role to play on the performance of the product, but yet we wanted to see if the globule size distribution varied, across different products. We saw that, D10 and D50, D90 values were all clustering. They were not really significantly different in both the types, and like metronidazole products as well as lidocaine products. Compare mineral oil and 3C in all the cases. They were very close to each other. Now let us see the drug distribution between the oil phase and aqueous phase. This was kind of expected because the drugs are highly water soluble. You see that most of the drug, 90% of the drug was found to be in the aqueous phase, both in case of lidocaine as well as in case of metronidazole cream. Again, there was no difference in the amount of drug present in the aqueous phase between a 3C and mineral oil product. Like, for example, lidocaine, the amount of drug present in the aqueous phase was 92%, and in case of mineral oil, it was about 90%. Likewise, in case of metronidazole, it was 93 for 3C cream and 91 for mineral oil, respectively. We subjected these cream products to in vitro release testing. The in vitro release rate is tabulated here for lidocaine and metronidazole cream, and if you see the numbers, they're all within a narrow range. Compare the in vitro release rate of drug from 3C and mineral oil products. They did not differ significantly in lidocaine cream products, as well as in case of metronidazole cream products. They were pretty close to each other. Now, we have completely eliminated any role of critical quality attributes on the performance of the better performance of the 3C product over the mineral oil, right? Because they were matching in terms of their critical quality attributes. Now, to what else can you attribute the enhanced permeation of drug from the 3C product to, other than the nature of the 3C to act as a chemical permeation enhancer? Again, regardless of the mechanism, we don't know what mechanism is responsible for such an enhanced permeation of drug from the topical cream products, but at least we can now confidently tell that 3C could act as a potential permeation enhancer for drugs like lidocaine and metronidazole. In conclusion, I would like to say that fatty alcohols are very important in preparation of topical cream products because they impact the quality attributes of the products. They play multiple functional roles in the topical cream products. The oil phase that you chose should be dependent on the physicochemical nature of the drug, because a good match in the physicochemical characteristics of the oil phase with the physicochemical nature of the API would result in optimal drug delivery. And from the experiments we did, we can confidently say that the Qualicream OD was found to be a promising permeation enhancer for a lipophilic drug like azelaic acid, and Qualicream 3C was found to be able to significantly enhance the permeation of hydrophilic molecules like lidocaine and metronidazole. Lastly, the most important slide, I would like to acknowledge the funding support from BASF. I also would like to thank BASF for all the lipidic excipients they supplied for the projects. And I thank Dr. Howard Maybach for being my mentor. I'm grateful to all my collaborators, research scientists, and graduate students for contributing to this project. A special thanks to Dr. Sam Rainey and Dr. Priyanka Ghosh from OGD of USFDA. And I also would like to acknowledge the grant support from USFDA. TPT, is Topical Products Testing LLC, is a small contract research facility that I manage in the incubation center of the University of Mississippi. I offer contract services to pharmaceutical companies. So I thank you all for patient listening, and we are open for taking questions now.
So thank you all for joining us today. Once again, Frank Romanski moderating the Q&A session that we have here. First, I'd like to thank Benjamin Goodyear and Dr. Murthy for their excellent presentations today. So just to remind everyone, you can use the Q&A function on the On24 platform to submit your questions, and we will do our best to get through as many of them as possible, and if not, we will follow up with each of you directly. So I'd like to start with a question for Benjamin Goodyear. Ben, you tested various industry-relevant cream formulations using dermatological drugs, but all of them have relatively low log p-values, kind of in the range of 1 to 3. Can you maybe share some of your plans to test the effect of permeation on drugs that may have a log p-value for APIs higher than 1.3 and maybe the 3 to 6 type range?
Perfect. So yes, thanks, Frank, for the question. And you know, to kind of reiterate, the log p-values that we've tested are, you know, under the range of log p of 1 to 3. So now that we know that the log p-values that typically chemical penetration enhancers need to be deployed is going to be outside of that 1 to 3 range, the experiments that we studied in this, you know, with Dr. Murthy were, you know, between 1.33 and I think it's like -1. But the higher log p-values actually might be more interesting to study, because we know more lipophilic drugs will be traveling through the stratum corneum, you know, lipid lamellar pathways. So if the lipid disordering is happening on the lipid lamellar pathways, we might actually see an even more increase in the flux and cumulative drug permeation.
Thank you, Ben.
So I guess, yeah.
How about a question now for Dr. Murthy? Dr. Murthy, you used the word evaporative metamorphosis during your presentation. Could you maybe shed a bit more light on this phenomenon and why it is important for topical formulation scientists? Just make sure you're not on mute, Dr. Murthy.
Yeah. Frank, thank you so much for the question. The permeation studies that we performed were done at infinite dose conditions. Generally, for evaluating the generic products or any type of topical products, we use the finite dose conditions. As per the FDA guidance, I think it's somewhere between 5 milligrams to 15 milligrams per centimeter square is defined as the finite dose condition. So that's supposed to be somewhat clinically relevant dose of the semisolid that you apply on the skin. So when you apply such a small dose, the evaporation of solvent, kicks off immediately, and then as the solvent evaporates, there are multiple things, multiple changes that occur in the formulation that is applied on the skin, like, maybe because of the evaporation of solvent, the drug concentration would increase, or if it's a poorly soluble drug, it would precipitate out, or the viscosity of the formulation might increase. Sometimes even the formulation would contract, and the active diffusion area would also change. So these changes are brought about because of the evaporation, and this whole phenomenon we term it as evaporative metamorphosis. So that is the whole reason why we performed the in vitro permeation testing in the two projects at infinite dose studies. We did not want all those factors associated with evaporative metamorphosis to influence our results, because our objective was to just investigate if these lipidic permeation enhancers would act as permeation enhancers or not.
Thank you, Dr. Murthy. So, Ben, why don't we start with you for this question? Do you think that fatty alcohols would work also as penetration enhancers, particularly at lower concentrations compared to that which was shared today, something in the range of 3 to 5%? Again, make sure you're not muted.
Oh, sorry, I was muted. So lipidic excipients, right? We typically use, various percentages, and these various percentages have different functionalities. What you've mentioned in this question is the 3 to 5% range. What we know is that we need to optimize the formulation with a chemical penetration enhancer, and this can actually be done using some computer-guided software. And I think this is something that, you definitely need to consider, because if you don't, you might be missing out on a great amount of cumulative drug permeation or flux increase. What I've noticed is that even using as much as 2 or 3% of Cream 3C in for hydrophilic drug permeation, I've actually seen very significant increases in flux. So as little as 2 to 3% will do you really well. But actually, if you use 3 to 5% or even up to 12%, you can actually get even more benefits, like some of the sensory aspects. I personally like rich feeling formulations, and this could actually be a way to kind of help with some of those sensory cues.
Thank you, Ben. So, Dr. Murthy, one of the really interesting slides that you showed was comparing the concentration of drug between the oil phases and the water phase. Could you share with us and the audience, how did you determine the concentration of azelaic acid in the various phases of the cream?
Frank, that's a great question. We looked at the drug distribution between the aqueous phase and the oil phase in all the creams, lidocaine, metronidazole, as well as azelaic acid. The way we generally do it is by subjecting the cream products to ultracentrifugation. Of course, every product is different. You have to develop the method. You have to optimize the speed and the duration of ultracentrifugation for the complete separation of the oil phase and the aqueous phase. So we subject the creams to ultracentrifugation, and then once the phases separate out, we aspirate the phases and look at the amount of drug present in them.
Thank you. So Ben, can you share with us if you're planning any future experiments to understand the mechanistic pathways of various dermal drug delivery using different pharmaceutical emollients like Cream? You showed some of the results today. Where are you going with this type of research?
Thanks, Frank. So this is actually something we're super fascinated by because we know that the lower log P value drugs are able to effectively be enhanced using these lipidic excipients. What if we actually use lipidic drugs? What would the effect be in that case? So I think this is kind of like an area of understanding the mechanism and the pathways, or the mechanistic pathways involved in the dermal drug delivery of these various lipophilic APIs. I think this would be our next chapter.
Thank you, Ben. I also noticed that we have a number of questions related to the slides for this presentation. The slide that's currently on the screen on your Un24 platform should have the contact information for each of us. So if you are interested in the slides, please reach out to us and we can have a discussion on that and be able to share portions of the presentation. Dr. Murthy, I want to ask if you could maybe describe a little bit more about the capabilities that you have at your research facilities at the TPT LLC, the Topical Product Testing Center.
Sure, thanks, Frank. Topical Products Testing LLC is a non-GLP type contract research organization. We offer contract services to pharmaceutical companies from the drug discovery sector. If there are any, if anybody is looking at new chemical entities as potential drug candidates for dermatological products, and if they want to look at the skin permeability for those molecules, we can do that. We have the capability. And we also offer contract services to generic product manufacturing as well as innovative product manufacturing companies as well. So anything that is related to topical products, characterization as well as evaluation, if anybody has a question, I would encourage them to contact us.
Thank you. So Ben, I noticed a couple of questions that had come in related to basically the drug being more soluble in the aqueous phase versus the oil phase. Again, questions about the log P of the type of drug. Would you be able to elaborate a little bit more about the permeation of various drugs depending on their inherent solubility in the system as well as the partition between the oil and the water phase?
Yeah, and that's a good point, Frank. I mean, I can't comment a whole lot here because, I mean, we only have a certain amount of resources to be able to do that. And this is kind of why I rely on the expertise of, Dr. Murthy and all the experts in the field. But it's something, I think maybe if there was a specific aspect in terms of solubility that we could answer in terms of being in the dispersed phase or inside the vehicle, where is the drug located? I mean, we need to know a few other things specific to the type of drug it is, because we know everything is very drug specific. If we take something that's more hydrophilic or more lipophilic, it will have an impact on the resulting microstructure of a formulation, and that microstructure could actually impact the dermal drug delivery. But I think we would need to know very specifically what your plan would be and what dosage form to help you answer that more specific.
Yeah, thank you, Ben. I think it comes down to these are very complex and dynamic and non-equilibrium systems. Each formulation is unique, and if you're curious on any specific examples, to reach out to us, to reach out to Ben and the global network that we have in the skin delivery platform to work on those types of questions. The last question for Dr. Murthy. In your experiments, was the donor cell occluded or not? And if not, can you try occlusion to stop evaporation? And can that check with finite dose, which would be more clinically relevant?
Thank you, Frank. It's a great point. Actually, I missed that point during my presentation. All the studies, in vitro release testing and in vitro permeation testing, studies that we performed here, we occluded the donor. We did occlude the donor. We had to prevent the evaporation. So we, like how we do in case of IVRT, we did in case of IVPT as well. We had occluded the donor.
Okay, perfect. So once again, I want to thank our speakers today, Benjamin Goodyear and Dr. Murthy. We really appreciate the outpouring of questions that we've had and also the very good participation in this. This will be available on demand as well if you want to share it with any of your colleagues that were unable to join. And also you have our contact information if you would like to follow up with us once again on any more specific questions or would like to chat with either Ben or Dr. Murthy on various technical questions. So once again, thank you. And we appreciate you taking the time to meet with us today. And please feel free to reach out to us again with any questions, samples, etc. Thank you.